Good morning or good afternoon, everyone, depending on where you are joining us from, and welcome to today's webinar. My name is Garrett from Business Review, and I will be your host today. It is our pleasure to have LTS with us today. We'll be presenting this webinar titled Transdermal Therapeutic Systems. Today's guest speaker is Dr. Marco Emgenbroich, Corporate Vice President, Head of Pharmaceutical Development at LTS. I'd like to welcome you to our webinar platform On24. You'll notice that this webinar is browser-based, so if you do disconnect for any reason, please just click on the link that you received via email to rejoin the session. In order to ask questions, you can send them in via the questions widget. Just type them into the box at the top left-hand corner of your screen and click submit. We'll allocate some time at the end of the session to address any questions or thoughts that you may have. Please use the yellow help widget if you require any assistance, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But now, please allow me to welcome Marco.
Yeah, thank you, Rick. Thank you, Garrett, for the introduction. And let's first welcome everybody in the audience from my end to our today's live webinar. Before we dive deeply into Transdermal Systems, let me quickly introduce you to the company LTS, Lumen Therapy Systems. So LTS is a mid-sized company located with the headquarters in Andernach in Germany, where I'm sitting right now and talking to you. LTS is about 1,200 people. And we are producing here in Germany the major part of our commercial products for the global market. And we have all R&D operations here as well in Germany. We have a U.S. subsidiary in West Corwin, New Jersey, where we produce mainly for the U.S. American markets. And we have an Asian representation sitting in Shanghai, taking care of our Asian partners. We are mainly developing and producing transdermal therapeutic systems, but also our thin films, and looking into new emerging technologies. Now let's dive into transdermal therapeutic systems, and let me give you firstly a very quick introductions in how patches would work and how formulations could look like. So this is a very easy schematic scheme on the human skin tissue. We have, with the transdermal patch, we have to overcome the uppermost barrier of the epidermis, the stratum corneum. And to achieve that, a few prerequisites have to be set so we can enable this drug permeation into the skin and through the skin. Number one is adhesion. A patch has to adhere during its application time, might it be from 1 day up to 7 days. And the adhesion should be at least 90% plus to enable the drug delivery through this application time. The second prerequisite is occlusion. So the transdermal patch has to create a minimal occlusion to entrap the moisture, the water in the skin to enable and enhance the permeation. Based on, after these two prerequisites, the known model of the NATME principle is also applied to the transdermal systems to ensure and enable the right pharmacokinetics. Over the last about 3-4 decades, a number of transdermal patches have been developed and commercialized into the market. And mainly the patches are not looking all alike. All sorts of different designs have been developed to meet the product profile, but especially to accommodate the problems and the needs of the API. So either the drugs could be volatile, for example, like nicotine, or pretty expensive, so you have to really save the API with a patch design. Of course, commercially driven, easy designs are most preferred, but it's not always the case and always possible. Let me quickly show you the two formulation designs, and starting with the very easy one, which is the drug and adhesive monolithic matrix system. So in this very most common design in the commercial markets, it consists of 3 layers, starting with the backing layer to protect the adhesive layer while applied to the skin. The second layer is a drug-containing adhesive matrix. And the third layer, a release liner, the protective liner, which has to be removed prior to the application of the transdermal patch. In this case, the adhesive layer has to fulfill several functions in the transdermal systems. Of course, number one, it has to enable the adhesion of the patch to the skin during the application time. But second, the adhesive, together with other functional excipients, have to dissolve the API or keep it in a stable physical-chemical status in the drug matrix. So it's really available from permeation. And lastly, of course, it has to release the drug substance into the skin to enable the constant delivery. Due to the chemistry and the nature of drug and adhesive matrix, usually less than 50% of the API can only be released into the skin. The majority of the patch the API stays in the patch and remains there. A more advanced technology being used in the transdermal systems are microreservoir systems. So this is a design where the API is not dissolved into the drug matrix, but it's more dispersed in a separate phase, either a solid inner phase or a liquid inner phase, to achieve the right solubility of the drug in the whole matrix systems, but mainly to target two points, two functions. Is number one is a high or maximized API utilization, and number two is an optimized flux profile, really to have a faster onset of action and a really increased flux, which at the end of the day makes the patch smaller and less drug can be used to enable the right dose. In general, the patch design is looking the same like the drug and adhesive, so we have a backend layer and a release liner with its similar functions. To dive a bit deeper into the microreservoir formulations, we have a scheme here which shows the design of these microreservoirs. So again, you see on the upper figure the backing film and the removable release liner. And in between, there is the matrix layer or the adhesive layer. And the adhesive itself is usually not very good solubilizing the API. So we create an inner sphere, can either be solid or liquid, where the API is dissolved in a stable under-saturation manner. So the patch can be stored very easily. And these spheres are, as the name is saying, in a micrometer range. So it should not have contact to the skin layer directly or to the outer layer directly to enable not a burst release, etc. On the bottom row, you see the function, what happens when you apply the patch to the skin. So first, the solvent in liquid microreservoir, the solvent in the inner sphere is permeating firstly into the skin. So it's a good penetrating molecule, which is going first into the skin, and by that it creates an oversaturation in the inner spheres, and the API is by this oversaturation activated and being driven into the skin. So this is a high drug diffusion created by supersaturation. With this technology, we can maximize and increase the flux, especially in the very early time points, and to achieve this before set task of a high API utilization, high drug flux. So now at that time point, we want to ask you a poll question, and for that I hand over to Garrett.
Thank you very much, Marco. So the first poll question today is, do you have any personal experience with transdermal drug delivery technologies? Yes, I have already been working on that intensively. Yes, but I have only basic knowledge. No, I haven't looked into it yet. What results are you expecting, Marco?
I would expect at least most of the audience people click on a yes. Whether they have actually worked on this or not, I have to see about that. But maybe we can convince more people to actually work on this.
Thank you very much. So now the audience have had a little time to vote. We'll move on to the answers. So, yes, I have already been working on that intensively is at 44%. Yes, but I have only basic knowledge at 36%. And no, I haven't looked into it yet at 20%. What do you make of these findings?
I think that's more or less what we already seen in the market, that a lot of people know transdermals, but not the majority have worked on this. Well, in this case, yes, but we're hoping to convince companies and other scientists to consider transdermal route as well. And maybe I can do so in the next part. Okay. So let's jump to the next chapter, and I would like to introduce you into a key importance which we think of transdermal therapeutic systems, because mainly the pharmacological advantages and therefore the benefits for patients and caregiver can be a driver for patch development. So here we see a list of driving reasons why a transdermal delivery should be considered. So there's no pre-systemic metabolism. We don't have no gastrointestinal passage. A transdermal patch can achieve a very controlled plasma level, even with drugs having a short half-life. A transdermal system allows a wide dose range with identical release properties just by simply having different patch sizes. There is no interaction with foods and no gender-related effects. And in the figure you see here on the right side, this is just a very schematic view. You see, so compared to an oral administration where you see the peak-trough ratio during each and every application time, a transdermal administration is much more constant over the application time. Even smooth one-day patches achieve a very stable plasma profile in this case. Now, when you're looking at the commercial products in the market over the last, say, 30 years, the commercially successful products, they all come along with one major pharmacological advantage, which is the key advantage for the patient, also for the caregiver to apply those patches. So it can be either, like starting with the hormone products, a reduced drug exposure, which is definitely lower than compared to tablets. It can have reduced side effects, and the patch can compensate short half-lives. But also, like for the pain medication and infusion like drug delivery is very important and change the game in a way. By going to a transdermal route, we can also overcome low bioavailability. And as mentioned before, a simple dose adjustments of the same laminate but different sizes. And we'll see that later in the manufacturing part. Now let's jump into a few examples where these, I don't see it right now, give me 1 second. One moment, I'm experiencing some black screen, white screen. So let's go into a few examples. Starting with the patch or the API oxybutynin. So this API, the key driver of having that patch is no first pass effect. Oxybutynin is used in the indication of an overactive bladder syndrome. And one key side effect of the oral dosage forms is mouth dryness by liver metabolite, the N-ethyloxybutynin. And as you see in the picture on the bottom right, the immediate release dosage form has a high plasma concentration of the metabolite NDEO, at about 20 nanograms per ml. And by moving into the transdermal route, this metabolite could have been reduced significantly. And as seen in the clinical trial, the side effects like dry mouth or constipation were similar only to the placebo level, which is a key advantage of the transdermal patch. A similar key advantage has been seen for the API rivastigmine. Also there, the reduction of side effects was one of the key drivers, just by circumventing the GI passage. Rivastigmine is used in the Alzheimer's dementia, and the capsules, which have been on the market for several years, there have been very often observed side effects like nausea and vomiting. And these major side effects caused a lot of patients to discontinue the treatment. It was pretty severe. And by moving to the transdermal patch, a late-stage clinical trial showed that the adverse events has been reduced compared to the capsules because they are GI-related about 3 times. So the transdermal treatment was very well tolerated, and the reduction of fluctuation was significant. So this change to the transdermal route was a key driver of the rivastigmine and the Exelon patch to be so successful in the market. And even after introduction of the patch, the sales of this product went up again, hopefully, possibly due to the reduction of side effects. Another example are, as said before, the infusion-like drug releases. This is a very good example where a transdermal system can be applied for drugs with a narrow therapeutic windows, like seen in the schematic here on the right side. And the key examples here are definitely opioids like buprenorphine or fentanyl. So by a transdermal delivery of several days in this case, we can achieve a permanent infusion-like drug absorption and a pretty good release control or optimal release control of this drug substance. These pain patches, so to say, are in the meanwhile really a standard of care and pain management on a global basis, but of course have to be handled very carefully so that patients are not misusing them. The last example I would like to give you is the API reticotine. And in this case, by moving into the transdermal patch, it was the overcoming of the low bioavailability, which was really insufficient, was achieved, and basically the API was rescued. Otherwise, this drug substance would have never been developed. So this is in the indication of Parkinson's and restless leg disease, the first and only ever developed NCE only for transdermal patch. So far, we have not seen another product coming onto the market where an NCE was developed only by a transdermal patch. This NUPRO patch uses a very efficient microreservoir matrix technology and is available in 6 strengths from 1 to 8 milligrams per 1 day. So we see the different flexibilities and opportunities which a patch allows are used here in this product. So in summary, the pharmacological advantages which we see for patients and caregivers are like rapid identification of the medication in case of emergency. So like emergency doctors can see if the patient is already on a pain medication, for example. The drug delivery can be terminated by a patch removal, somewhat terminated, of course. Still, there is some drug in the skin, which later on is still distributed. But compared to all dosage forms, especially controlled or slow-release dosage forms, at that time already we still stopped the major delivery of the API. A transdermal patch can achieve a higher level of patient compliance and allows a better monitoring, especially for caregivers and for patients which are in daily care hospitals, so it can be easily monitored externally, so to say. A patch is independent from food intake, and the ability to swallow, nausea, vomiting, or diarrhea. The patch is non-invasive, and it reduces, as said before, the side effects. Now let's go to the manufacturing of transdermal patches, which is pretty unique compared to other dosage forms like tablets, for example. So we usually differentiate between 4 different unit operations for the transdermal patch manufacturing. So it starts with mass preparation, we go to coating, drying, and lamination. The next part is slitting, and at the end, punching and pouching. And I would like to go a bit more deeper into these 4 steps. Starting with mass preparation. As seen before, the easiest drug and adhesive matrix. So for a product, it can be as simple as it sounds. We only have to mix the API and the other excipients into an adhesive solution. The adhesives usually come in organic solvents. We can add, depending on the formulation, other polymers which can have film-forming properties or stabilizing properties. We can add softeners, enhancers, or some stabilizers like antioxidants, for example. The commercial batch size can easily be in the range of 100 to 800 kilograms, depending on the product, of course, and usually can be mixed, sometimes easily in a stainless steel vessel. But very often, the mass preparation also contains a bit more sophisticated process steps like high-speed homogenization, heating or cooling steps, or even premix solutions which can be added stepwise to accommodate the needs of the product and more complicated like emulsion or dispersion types of formulation. In the next step, we bring this adhesive vessel, so to say, into the coating line. And here you see the scheme of a mid-sized coating line with 5 drying channels, drying zones. And on the bottom left, the adhesive is transferred from the vessel into the coating head. Here you see a 2-row coater. And this adhesive is then coated onto the release liner, which is used very often as a coating substrate. So, and the coated adhesive mass, still wet at that stage, is then transferred into the drying oven. And there the organic solvents are removed by a distinct drying profile. So at the end of the zone, the adhesive layer is coming out dried, solvent-free. We can achieve the limits of the ICH guidelines, but hopefully mostly we are significantly below that to create a stable product. And the last step, we laminate the backing film here to this adhesive layer. So we achieve at the end a 3-layer laminate, which I've shown you before in the design of the drug and adhesive patch, consisting of the release liner, the adhesive layer, and the backing film. Now these are rolled up into so-called master rolls, They can be small, like starting with 20 or 30 centimeters, up to 1,60 meters easily. These master rolls are then transferred to the next step, where slitting. So it's also pretty unique for pharmaceutical manufacturing. So this master roll is then slit down by a knife technology into different narrow rolls. So these narrow rolls are already predetermining the sizes, the width of the patch, so either one or two or multiple patches next to each other, which can be produced later on in a subsequent step. These narrow rolls can be stored as an intermediate and then used for the different territories, different countries in the next conversion step. So the conversion lines, they can be looking very, very different in its design. On the left side, you see a picture of one line commercially used. This process, to be seen from right to left, starts with the unwinding of the laminate, the narrow roll. Then we punch in the application aid. It can be a straight cut or wave-shaped cut. And each patch is imprinted with the dose and the name of the drug product. And the patch is die cut out. So the rest remaining around the trellis is still API containing, is by then removed. And the final patch is cut and then transferred into the pouch material, and later on sealed, closely sealed into a sachet. So each individual patch in a single pouch, the sachet is then packaged later on. Subsequent processes like secondary packaging, etc., also have to be fulfilled to meet the local requirements. But these are the key steps which are responsible for the manufacturing of transdermal systems. So at that time, we also have another poll question for our audience, and for that I hand over to Garrett.
Thank you very much. Poll question is, from your personal experience, do you think that transdermal drug delivery is still an attractive technology? Yes? No? I don't know. What results are you expecting from this?
I mean, that's a key question which we are also asking ourselves. But as you might know, we think, of course, it's still an attractive technology, and I hope that in the last part of the presentation, I can convince the audience that it actually is.
Well, let's find out everyone's view. So, 95.3% for yes, 2.3% for no, and 2.3% for I don't know.
I'm somewhat overwhelmed already by the positive answers, which is good. I mean, that's the reason why we are here today. So let's jump in the next part of our webinar. And I would like firstly to jump into the global market to see whether it's still attractive also from a commercial perspective. So over the last about two decades, we have seen a number of products hitting the markets, and each product really also determines the market size. So starting in the early years, before the years 2000, where we have the fentanyl patch, the duragesic on the market, nitroglycerin patches, the hormone patches and the replacement therapy, but also the nicotine products. And after that, we see the key products which have driven the market are like the buprenorphine powered products, Transdermal and Norseman, which have been launched in the 2000s. Of course, the EFRA contraceptive patch already made a great impact. But on the other hand, then the market share go down after generic entry for the fentanyls in the mid of 2000s, which now is a completely generic market. In the later years, we've seen the Nupro and Exelon launches on a global basis, which also have driven the sales significantly. And when we're looking at the numbers on the next slide, you see the green curves are the revenues over the last about 2 decades, going slightly up to a peak at 2014, roughly $6 billion. And since that time, it's slightly going down, and downwards is basically because of the generic entries of a few of these big products. But you can also see the dip in the year 2005 in terms of sales, which was the entry of the fentanyl generics. In terms of the quantity and number of billion patches, we see a slight decrease over the last year. So even the sales are pretty high. We're now in the range of somewhat around 1.7, 1.8 billion patches. And we are now waiting for new products to hit the market, not to change this view again, because this is what we definitely hope to see in the future. So now we come to the key part of our webinar. I would like to show you some case studies, which are, I think, very important from a commercial perspective and from the view for future development to what is possible to accommodate the needs. So the first case study is about a Japan-specific product. So we see more and more requirements coming from different territories, different countries, and so the necessity to develop a specific formulation to meet these requirements. Especially a product differentiation might be necessary for adhesion properties. So different markets and different patient populations, but especially different local habits, can trigger such kind of differentiation. So the one patch for a global market might not be the solution for the future. So we have seen from commercial products that the adhesion or the perception of the adhesion can trigger different complaints. Where a patch, due to a strong adhesion, can cause skin irritation in Asian markets, the very same patch can cause the complaints of lack of adhesion in Western countries like US and Europe as well, but other countries. So there might be a differentiation necessary to accommodate that. We have also seen already significant formulation changes triggered by a local habit in the fentanyl market in Japan. So moving from a 3-day patch now to a 1-day transdermal systems in Japan within the last 5 years, which was mainly triggered by the habit of a daily hot bath. So this basically flipped the patch market in Japan. Up to 90% are now on the 1-day patch. Recently, this trend has been applied to one big product in Japan, to the Exelon and the Rivastach. So when you see on the right side, in terms of number of patches being sold only in Japan, we are hitting end of 2018 already almost 15 million patches. So this is a very important market with an important product. So the product which was on the market so far was based on silicone and acrylic pressure-sensitive adhesives. They're really optimized for a very good adhesion profile on a global basis. But this product has caused the reports for dermal syndromes at the application sites, like redness or pruritus. And these side effects on the skin irritations can cause possibly a discontinuation of the treatment using the patch and causing patients moving to other dosage forms or other APIs. So to accommodate these possible problems, a new more skin-friendly formulation was developed, and this product was actually already launched mid this year, 2019. And I'm looking forward to see whether this adjustment of the product formulation will be translated in a positive market feedback. Now let's jump to a second case study, which is a bit different to that, and we're looking at a more rational approach of a drug development or a drug product development from the lab into the clinic. And we're using the example ASENAPINE as a multi-day patch here for this case study. So the way LTS approaches development is the separate steps in looking into the feasibility of an API to be delivered transdermally. And the first step is our trace database for first in silico assessment. The trace is an acronym for Transdermal API Candidate Evaluation, where we collect all sorts of physical chemical parameters, put them into a database, into an algorithm, and we receive as feedback a kind of scoring from less favorable to favorable. And the example or the outcome for the API is ASENAPINE, which is used in bipolar disorders or schizophrenia in adults, was pretty positive, as you can see here in this figure. So the score is in a positive range. So usually, as a first just paper, computer exercise based on this database, the first hurdle is already done. So we now go into the second step of the feasibility, which is basically skin permeation studies in different in vitro diffusion cell models using a front cell type of permeation studies, either on human skin or animal skin models, like, for example, mini pig skin. And here you see just pictures on the left side, a patch applied onto a skin sample on the front cell, but pictures also from automated front cell systems that we're using within our R&D department. Now, as a result for the permeation studies of asenapine of different formulations in mini-pig skin, you see here on the bottom left in the figure the flux data in microgram per square centimeters an hour over the time of 72 hours. So the different formulations are all significantly above 10 micrograms per square centimeter an hour, and this is what the target range which we try to achieve in this. By translating this into the in vivo situation using the same mini-pig species in a preclinical study, we see a nice translation of these profiles into the plasma profiles using the mini-pigs. And the next step, or in parallel, we already performed human skin permeation studies using excised surgical skin, so not human cadaver skin for this study. And we're achieving a very similar flux profile, slightly lower than the mini-pig skin, which is in this case it's expected somehow because the skin properties are not identical between human skin, of course, and mini-pig, but somewhat different. And here we experience slightly lower flux, which was expected. So this positive in vitro data plus positive preclinical data, it made the team to select the right candidate for a clinical trial based on this data. But before we go into the clinical trial, we're using something new. And we are looking at a graph which is created by a new LTS proprietary software. It's a new type of trace evaluation for in vivo modeling of a plasma profile. So here we see just simply the computer modeled PK profile based on all sorts of in vitro and preclinical data set. We have to add into this software. So we see the blood level coming out of this modulation for different application times. So here we see the first application period, about 3.5 days, gives a blood level at about 2 to 3 nanograms per milliliter. The subsequent applications, they end up in the blood level between about right about below 3 up to almost 4 nanograms per milliliter. So remember this picture for the first application, plus for the repetition of the application, because we will see something very similar later. So now we went into a very first phase 1 clinical study, a bioavailability study in comparison with the reference product, the sublingual tablet Cycrest. So here we see 2 formulations being used in a clinical trial for 3.5 days. So ending up in the plasma range right below 2 nanograms per ml up to 3 nanograms per milliliter. This is what we have already been predicted by our tool. And we're now using this, so we have a successful translation of our previous in vitro data and the preclinical data into human trials. Now we're using those data, and we're going one step further, we do a PK modeling. So we're simulating the plasma concentrations of multiple dose administrations based on the real clinical data. And now again, you see in the red curves here the reference product, the Cycrest, going into a steady state after multi-applications. And then if you're looking especially at the gray curve for the test 2 product, we see at the first application, within the first 3.5 days, we are about in the same range of 2 to about 3 nanograms per ml. And once we go into steady state, so to say, in the second and third application especially, we are in a range of 3 to 4 nanograms. So we see a very good fit between the prediction of the plasma profile we have done with the trace in vivo modeling and the PK modeling based on the clinical data. So this new type of software we are now using opens up a new type of development. So we can de-risk a project by looking into potential plasma profiles beforehand, before we actually go into the clinic. And so this is used to have either a quick decision or a quick kill of projects, and to focus on those which definitely show the most success possibly. Now, moving away from the passive transdermal delivery. Now we're looking here at our hedgehog, which represents the new type of microarray patches, which we like to show you. This is something which we think is very important for the future to deliver different types of drug into the skin. This development of these new development technologies are done in our IAS division for innovative injection systems. So this is a separate unit at LTS taking care of these new delivery technologies. Within the IAS department, we have an R&D unit including a GMP suite for manufacturing of clinical trial supplies for early clinical phases. And we have a manufacturing license definitely for making these microarray patches for clinical trials. This unit is set up to develop devices, formulations, and all sorts of analytical methods. What are microarray patches for? This is what we think the next generation of transdermal or intradermal delivery. It's suitable for biologicals, vaccines, or small molecules which cannot be delivered by the standard passive route because of their physical chemical properties, or they're too large, or the dose is too high. We expect fewer side effects to the transdermal or intradermal administration, and there's definitely a lower risk of needle injuries and any cross-infections compared to any kind of intradermal needle, for example. We can also potentially improve bioavailability and therapeutic and cost benefits. So how does this work? With the microneedle technology, our MAPS, we would like to overcome the stratum corneum, the outermost barrier of the skin. And this can be achieved by needles. In this case, these are polymeric needles, dissolvable needles, in a range of 100 to 1000 micrometers. So these needles deliver the drug substance or the molecules right in the epidermal region, for example, where the immune competent cells are located, like Langerhans or dendritic cells. There is no stimulation of nerve cells or no harm of blood vessels, since these needles are not penetrating deep enough to do so. So in this picture we see a scheme of these dissolvable microneedles, so containing of a polymer matrix which creates the needles and the drug substance, the API, being embedded into the polymer needles. And this example shows an immediate release dissolution of a MAP array. And when you see on the left side the four pictures, starting in the top left, where there's a green circle, you see a microneedle sticking in a skin model. And within the time going from the top left to the bottom right, within several minutes, in this case 7 minutes, this picture, this needle dissolves in the skin and releases the API right away. These pictures are created by optical coherence tomography right in the skin model, actually. On the bottom right, you see another example being created where the base plates are created of an excipient without API, and only in the tip we see the API created with a red dye in this example, which you see dissolving within time. So at the end, I would like to summarize what a transdermal delivery and accommodating technologies around can show and can achieve. So we have seen transdermal patches already been proven options to improve bioavailability, like with a Nupro patch. Significantly, we can avoid first pass effects, a major driver to go to a patch as well. We can achieve constant plasma profiles. This is one of the key elements for the transdermal delivery, and reduce side effects. So overall, the development of a transdermal drug product should be considered for first-line drug delivery forms and not just a line extension, but of course also for line extension or better generics. It's a key option which can be used in future developments. So at that stage, I would like to thank you for your attention, and I can only invite you to explore with us further and create new products for the market. And for the Q&A session, I will hand over to Garrett again. Thanks.
Thank you very much. Just a reminder for the audience, in order to ask questions, you can send them in via the questions widget. Just type them into the box at the top left-hand corner of your screen and click submit. I already have a few for you here. The first question is, how long does a patient typically need to wear the patch?
One patch application time is usually either between 1 day to up to 7 days. So in the products which are on the market, either 1-day patches, 3 or 3.5, 4-day patches, or bi-weekly basically, or a 1-week patch. So, but in total, of course, the patient usually, since our chronic diseases, are wearing the patches or a patch every day.
Thank you very much. I have another one here for you. What are drawbacks of TTS?
Well, that's a good point. First, there are certain limitations in terms of drug dosing. So you can only deliver a certain amount of API through the skin. It's a barrier by itself. And the API should not have a significant skin irritation, because this is also something maybe to be considered a main problem with a few of the products as well on the market. Irritation caused by the API, not by the adhesives. I think products which are on the market are usually considered as pretty well, as long as they adhere good during the whole application time. I think that's also a key drawback. Adhesion on a global basis, because between the cold regions, northern regions, and high humid regions, there's always a key difference of skins, etc., which is really a major challenge for the product. Yeah, about that.
Thank you very much. Just a reminder, you can still submit questions via the Q&A widget in the top left-hand corner of your screen. I have another one here for you. Which polymers are you using for the preparation of the adhesive pads?
I think you're meaning adhesives in general. I mean, in terms of polymers in general, we're using either pharmaceutical grade adhesives, which nowadays would be, I would say, like acrylics, silicone adhesives would be the major portion of the products which we are producing right now. But also PRBs could be used for polyisobutylene, and SIS-based adhesive systems like SIS, like styrene, isoprene, styrene copolymers, block copolymers, which coming in pharmaceutical grade. So these are the major categories of adhesives we're using. On top of this, we're using several kind of pharmaceutical grade polymers as film forming polymers, or which can dissolve the API in a way. And this can be all sorts of polymers like PVP, hydrogids, etc., which are out there in the market and known in literature as well.
Thank you. Next question is, how long do you expect microneedle patches to be applied on the skin compared to regular patches?
That's a good one, since it's not in my exact area and division here, but I would say the products which we're currently looking at are more short-term applications, maybe a few hours, but not compared to other passive transdermal patches where you have a constant application. So it really replaces a shot, a single injections. But this is something for the future. There might be other options which can be accommodated by these products. So right now we are focusing on a few of these examples, but I think there's no limits which can be done.
Thank you. Don't forget you can still ask questions via the Q&A widget in the top left-hand corner of your screen. I have another one here for you. Which drugs are most suitable for TDDS, and how do you identify them?
I think, there are several parameters also out in literature which are pretty well known. In a certain way, we have to have some lipophilicity. Molecular weight should not be too high, and it should not be too basic, for example, and have some minimal solubility. And how do we identify them? And this is, one step is of course our trace tool, where we put all sorts of parameters in there. But in terms of limitations, I think APIs which are too hydrophilic or too lipophilic are not suitable. So that they either, even if they can go into the skin, they are stuck and cannot be penetrated further, or if they are too hydrophilic, they will actually not overcome the skin barrier. But if there's any interest in how we can evaluate the drug substance further, we're happy to assist that.
Thank you. Question is, what is the main market for TDS products? US, Europe, Asia?
Good point. So I'm just looking at my colleague sitting next to me. Thank you. In terms of sales, I think the U.S. market is still the most important market, but we see a lot of interest coming from Asia on these dosage forms. It appears to be that especially in Japan, but also in China, there's a great interest in transdermal patches and this delivery route. So this might change in the future, and yeah, see about that.
Thank you very much. Do you already have experience with immunotherapy?
In a way, yes. I can only say. Sorry. I mean, we have performed one clinical trial. I mean, this is public on a vaccine with microneedles.
Thank you. I have another question here for you. Switch from 3-day to 1-day fentanyl TTS. Doesn't this lead to an even higher amount of fentanyl remaining in patches / discarded?
No, not necessarily. I mean, the content of the API in a single patch was reduced significantly, moving from a 3-day patch formulation to a 1-day patch. So that at the end of the application time, the remaining API in a 3-day patch is still kind of a lethal dose. So, which is about, depending on the formulation, 30 to 50% of the drug substance. In the 1-day patch is about the same. It's about 50% remains in the patch, but it's still only half a daily dose, which is still in there. So from that perspective, in terms of misuse, it might be even better to move from a 3-day patch to a 1-day patch. I think overall, in terms of waste, out the back of my head, the total amount of API being used for 3 1-day patches is slightly higher than for 1 3-day patch. About that.
Thank you. A member of the audience says, thank you for these specific cases for Japan and China. When you look at those markets, how is the prevalence of TTS there?
I think in Japan, transdermal systems or patches in general are highly preferred. It's a very well accepted dosage forms in Japan. In China, there's only few number of products already on the market. So we don't have a broad experience yet by transdermal products, but we think that in general in the future, as soon as also the market evolves and more patients can also afford these dosage forms, also Chinese market can be of highly interested, and also the acceptance will be pretty good.
Thank you everyone for your questions so far. Don't forget you can still submit them in the top left-hand corner of your screen. I have another one here for you. What are typical adhesive tests to consider during the screening of formulation?
That's a good point. I mean, within which is known also from USP perspective, there's a number of analytical tests which can be done, which are typical physical testing, like peel adhesion, tag, or shear force. These are usually the standard QC tests which to be considered for the development of a patch. Of course, you have to ensure that the release liner shear force is also low, so a patient can always remove the release liner, the protective liner. But in terms of in vivo, that's also a holy grail. How do you test the adhesion of a patch? And before going into clinical trial, that's something, I mean, which is also a whole, I would say, kind of trade secret for a lot of companies, how to do that. At the end of the day, everybody has to perform an adhesion study in a clinical setting to convince everybody that the adhesion is the right way.
Thank you very much. Did you see new API coming, or the trend is more to develop generic products?
I see more the trend coming into new APIs, since generic transdermals, so generic versions of the current patches, I think this will be over soon, since most of the products will be generic in the different markets within the next years. So there's might be still a trend going on, but I hope, and this is also what we see, that companies are looking more and more into bringing new APIs or existing APIs already in other dosage forms into the transdermal field by addressing a definite specific medical need for patients and position these products for very specific indications and patient populations, and using the advantages, as mentioned before, of a transdermal patch for these new APIs.
Thank you very much. I have another one here for you. How do you overcome poor API solubility in TTS?
That's a good point, and I'll say somewhat of this is our trade secret. I cannot say publicly, but we're using all sorts of technology to put this into our transdermal patches. And if there's a specific interest into going a bit more deeper, I would be happy to discuss this separately.
Thank you very much. If your question isn't got to today, don't worry, the team will be back to you at a later date. I have another one here for you though. Is it possible to do in vitro release tests on synthetic membranes?
Yes, it's possible, I can say. I mean, the normal in vitro release testings are done without synthetic membranes, using different types of apparatus setups. But synthetic membranes can be used as a replacement for a skin model in a front cell type of setup. But of course, it has certain limitations, since the effect of a permeation enhancer will not be seen on a synthetic membrane. But if there's only a passive delivery just of the API itself, there's no other functional excipients which is modulating the skin, this could be an option, yes.
Thank you. Is a transdermal patch a packaging material or a medical device from a regulatory point of view?
Neither nor. So the whole transdermal patch, from our today's point of view, is a pharmaceutical product. In the US, there's a certain consideration of as a combination product. So the patch which is staying outside can be handled in a way as a device, but it's definitely the majority of the countries on a global basis, a transdermal patch is in its total design, it's a pharmaceutical product.
Thank you. What about in vitro permeation studies with APIs very insoluble in water and permeation media?
That's a good one, actually. It's a main challenge to identify the right acceptor medium, and I might not be the absolute experts for this, but I would invite you to send me an email separately on this, and I will get you in touch with our analytical experts on this, and to assist on that. I've seen where the question was coming from, so you're invited to ask that.
I have another one here for you. Is there any farm-grade PIB?
It's a good point. I mean, the material we're using, well, to be honest, I'm not absolutely sure about the quality grade of that material. I would have to skip that question and we get back to you separately.
What is better, a 1-day patch or a 7-day patch?
This is kind of marketing believing, I would say. From a development perspective, also from the pharmacokinetic perspective, I would definitely prefer a 1-day patch. If you're looking at steady-state PK profiles, a 1-day patch always achieves a much better profile than a 7-day patch. On the other hand, a 7-day product is pretty convenient for patients, depending on the patient population indication. This could be also an important driver. As soon as you deliver pretty constantly above a certain threshold, a significant amount of API. So this is really something which has to be considered at the beginning of a development, and which is the main target clinically, which has to be driven. It should not only be a marketing point, because a skin never looks good after a 7-day patch application. So this is also something to consider in terms of patient compliance, patients' adherence to the treatment, and the willingness to accept a transdermal product. Also from the caregiver perspective, I think an everyday product to monitor is definitely beneficial than if we have even numbers and so on. And we see feedback from clinics where this is also pretty well accepted.
Thank you very much. We have time for a few more questions. Can you comment on API stability in TDS versus, for instance, normal oral drugs? Do you see any difference?
Well, principally, there's not much difference compared when you take all sorts of oral drugs. Compared to a normal oral tablet, compressed tablet, as a solid dosage form, of course, a transdermal system is more like semi-solid, and the API should be dissolved in the matrix. So there could be some stability issues coming along with the type of formulation, but there are options to accommodate these problems, like inert packaging, addition of stabilizers, etc. So this is usually been done if an API is not stable enough by itself. So, but if we're looking at similar formulations or liquid gels, liquid formulations for oral dosage forms, I would say it is pretty much the same. One key difference might be is the processing and the coating part. So if an API is not surviving the process by itself, this could be a limitation.
Thank you. I have another question here for you. How much drug can be delivered per day?
I would say the normal products we're having are maybe up to, the highest delivery per day is the nicotine product with 21 milligrams, and its largest dose, and this already gives a certain limitations for the different APIs. And it really depends on the API's properties, whether we can achieve only several hundred microgram delivery per day or up to this 20 milligrams. So for good permeable drugs, we would be on the upper end. Most of the products would be in the range of several milligrams per day.
Have you ever tested your patches in allergic treatment?
Not that I know of. I don't get the question why allergic treatment of local allergies or systemic. I don't know yet, but no, I have to pass on this.
I have another question here for you. Why are there not more NCEs rescued by transdermal delivery?
That's a good point. That's what we are looking at, and of course, a lot of our competitors as well. That pharma companies which have a very interesting API in their hand, and the normal oral delivery route is not feasible due to a low bioavailability. And if these APIs have the right physical chemical properties to be delivered transdermally, that would be a definitely interesting choice to go to a transdermal route if the profile which we can achieve in terms of a PK profile are suitable for the indication. Why not more? I think that's also something that a lot of companies might rather go back to change the chemistry than starting a transdermal program because it's not within their first area of choice. Hopefully this might change in the future and the transdermal route is more established, so to say.
Thank you very much. We have time for one final question. That question is, how do you prevent API crystal formation in the adhesive matrix?
Yeah. I would say that's the formulator's expertise to find the right balance between a good patch performance and not to be oversaturated or metastable so it can recrystallize over time. And I think with our new types of formulations like micro reservoirs, etc., we have the technologies in our hand to do so, but this is really something, I would say, the magic behind the formulation. Can't say much more, I would say.
So that brings the Q&A section to a close, and that just leaves me to thank Marco for what was a great presentation, and to LTS for sponsoring this session. To the attendees, you'll receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. We look forward to sharing further webinars with you, so please do keep an eye out on the website just mentioned, and follow us on Twitter @BRWebinars for daily updates, and join our LinkedIn group, Business Review Webinars. Thank you once again, and I hope you all have a nice day.